afternoon and welcome to this session at ESOF 2020. The two speakers are coming on the stage, okay? So, my name is Beatrice Mautino. I am a science writer and science communicator with a background in biotechnology. And today, here in presence and online, of course, we will speak about the evolution of genetics from genes to genomes, as the title of this session, and the relationships between science, technology, and policy. Let me introduce you our speakers. They are three experts coming from three different fields. One of them, Michele Morgante, is literally from the field because he's a geneticist, and he works on the genetic of grapevine, and he is a full professor of genetics at the University of Udine and the scientific director of the Institute of Applied Genomics. He works, as I said, on the genetics of grapevine, and is particularly active in the Italian and European debate about the genetic manipulation of plants from GMOs to the new breeding techniques. Then we have Mauro Giacca, professor of cardiovascular sciences at King's College in London and professor of molecular biotechnology here at the University of Trieste. Mauro Giacca is an expert in gene therapy and in the development of nanomedicines for cardiovascular application, and he is also very active in science popularization. He writes a weekly column on Il Piccolo, that is the local newspaper, and so I know that he has a lot of fun here in Trieste. And then virtually from the USA, we have Sheila Jasnoff. I don't know if she has with us. She is a professor of science and technology studies at the Harvard Kennedy School. She is a pioneer in her field. She is author and editor of hundreds of papers and more than 15 books. And her work explores the role of science and technology in law, in politics, and in the politics of modern democracies. So, let's start. We have 1 hour and a half. For the first round, I would like to ask to our speakers to help us to understand the situation. If we look at the history of genetics, we could make some turning points, some moments that revolutionized the discipline. The first one that comes to my mind is in mid-19th century when scientists like Charles Darwin, Wallace, and Gregor Mendel try to understand how life works and try to answer to questions like who we are and what determines our identity and our diversity, but at that time they had no knowledge of what the underlying genetic material could be. The second step that revolutionized the discipline occurred in the mid-20th century with the discovery of the structure of DNA and the techniques to cut and paste and to join DNAs coming from different sources with the techniques called recombinant DNA that give the opportunity to redesign nature in a sort of way. And in this period, we also had the first discussions at the level of the public opinion, but also in the scientific community, about the opportunity to manipulate life, to manipulate plants, animals, but also humans. So we arrive to these days, and I arrive also to my question, because today we assist to an incredible acceleration of genetics, as we said, from genes to genomes. So we are facing a totally different landscape from the scientific side, but also from the ethical, social, and political side. So what I ask to our speaker for this first round is to help us to frame this new landscape in order to understand which are the most important issues from their different point of view. At the end, we will open for questions from the audience here in Trieste and also online. I have a tablet where I can read the questions, so if you have any question, write it or raise your hand if you are here in Trieste, and then we will answer. So to start, Michele Morgante, you are the geneticist on the stage. So would you like to start and help us to understand the evolution of your discipline and try to help us to know this landscape, this new landscape? Michele.
Well, thanks a lot for the opportunity to talk here. I want to thank the organizing committee for setting up this interesting panel. And I want to start by giving two simple figures. The two figures are 100,000 and 1.5 million. 1.5 million is the increase in sequencing capacity, which has occurred over the last 15 years. 100,000 is the decrease in cost on a per unit basis in sequencing that has occurred over the last 15 years. So the combination of these two figures, which can be multiplied with each other, tells you how much the landscape has changed. And it has been a technological revolution, much more than a scientific revolution. And what I want to talk about in the next few minutes is the relationship between science and technology, and how these has changed our way of looking at genes and genomes. Why has this technological revolution been so important for our understanding of what determines the identity of living beings? Because their identity is written in a very simple code made of 4 letters, A, C, G, and T, which are the building blocks of the DNA sequence that makes up our genome. Today, we have an unprecedented capacity to read those building blocks. I would argue that as a science, we have not been able to follow up on the technological advancements, and in addition to determining the structure of the sequence, we have not been equally able to determine the significance, the meaning of that sequence. So how that sequence determines the visible appearance of the living beings. The relationship between technology and science in genetics has changed a lot over time. When genetics started in 1866 with the work of Gregor Mendel, genetics was purely a formal science. Mendel did not have any idea about DNA. He did not even have any idea about genes. He thought that there were a huge number of particles that determined the inheritance of traits from one generation to the other. That concept has been changing over time. Genetics, though, continued to be a purely formal science for a long time, because it took almost a century to determine what was actually the physical material that was determining those phenomena that were being observed at the visible level. And it was only in 1952 when it was definitively demonstrated that DNA was the genetic material. And in the following year, with the work of Watson and Crick in 1953, the structure of DNA was elucidated. But even though we knew what the structure was, we knew that DNA was a double-stranded molecule with 2 complementary strands, with some fixed rules of pairing between the 4 ACGT bases. We still did not have a technology to support us because we still did not know how to decipher the sequence of A, C, G, and T. And it took another 25 years to get there. In 1972, we finally learned to manipulate DNA with the development of cloning technologies that led to the era of recombinant DNA that Beatrice was mentioning. And it was only in 1977 when finally 2 different groups, one led by Wally Gilbert, the other one led by Fred Sanger, developed the first DNA sequencing technologies. Finally, genetics was able to look into the fundamental blocks that were determining the phenomena that far, in 110 years, it had only been able to study at the formal level. So that was the first revolution, clearly. Sequencing, though, was a long and painful process at the time. But still, there were some visionary scientists that at the beginning of the 90s started to think that it was time for us to embark into a major project, and that was the Human Genome Project. It was a project that was justified on the basis of not so much understanding our identity as human beings, but rather as a project that could provide useful applications. I think it should have not been like that. I think understanding what makes us what we are is important enough by itself not to need any other justification. From this point of view, I'm envious of our colleagues that work in high-energy physics that don't have to provide any justification as to why they spend billions of euros to understand the first milliseconds after the Big Bang. I think deciphering our identity is at least equally important, and I think it doesn't need any further justification. But still, that project took off. Initially, it was thought that in order for the project to be completed, there should have been another technological revolution. Nobody was convinced that the existing sequencing technology developed by Fred Sanger would have allowed us to get where we needed to get to decipher the 3 billion bases that make up our genome. But instead, when in 2001 the human genome was sequenced, it was sequenced with the existing technology. And it was thanks to a number of incremental improvements on the existing technology that that very important goal was accomplished. I think in 2001, we finally reached a point where technology and science were more or less in sync. We had finally identified the sequence of the 21,000 or so genes that make up our genome, and we could start to work with them. After 2001, both technology and science kept moving on. Technology, in terms of sequencing, developed enormously. And as I mentioned before, there was a revolution that led to the development of completely new sequencing technologies, which go under a collective name of next-generation sequencing technologies, that have transformed what used to be a serial process into a parallel process. As in any other industrial process, when you're able to parallelize what was previously serial, you achieve huge increases in throughput and huge decreases in cost. And that was also the case for sequencing. So that's how, starting in 2004, more or less, we got to where we are today. So we have a sequencing technology that today gives us access to the sequence of any living being at extremely low costs and extremely high throughput. has that capacity to decipher the sequence being paralleled by a corresponding capacity to understand the meaning of the sequences that we are producing? My opinion is that it has not. And today we are at the opposite side of the situation of where we were in 1866. In 1866, we had a lot of science and very little technology or no technology. Today we have a lot of technology and a science which is still not capable of utilizing entirely the huge amount of information, of data that we are producing. Why is that so? I think it's because genetics has lost the capacity of abstraction. The work of the pioneers in genetics was characterized by being largely theoretical initially, and then find its proofs in experimental work. With the luxury of being able to produce a lot of experimental data, we have fallen into the opposite problem. We think that the data itself is going to be self-explanatory, and we think we are going to derive rules and laws from looking at the data, but that's not the case. And here again, I want to go back to how physics works. If you think about high-energy physics, it works the other way around. So first they develop theories, and then they think about experiments that are needed to confirm those theories. If you think about the Higgs boson story, that's how it went. The theory was developed a long time ago, and it took a long time to perform the experiments that confirmed that the theory was indeed correct. In biology, and especially in genetics today, we do not have the ability, and perhaps we are not even used to, developing theories and then perform experiments that should confirm those theories. And I think this is really limiting our ability to make full use of all the information that we produce. We thought that perhaps the function of the genome would have been directly derived from its structure, but that's proving not to be the case. It's not sufficient to have the complete sequence of the human genome to understand why we are what we are, or why we suffer from diseases that we would like not to suffer from. How can we go past this? Well, definitely we need a change in paradigm. We probably need to educate a new generation of biologists. Biology today needs to be much more interdisciplinary than it used to. In order to make full use of all the genomic information that is available to us today, we need not only biological concepts, we need mathematical concepts, we need concepts in informatics, we need an important discipline that has arisen in the last 30 years, which is called bioinformatics. And we need to make biology and genetics much less an observational science and much more of a predictive science. Biology is largely based on correlations, but correlation is not causation. We need to be able to identify cause-effect relationships in order to be able to describe biological phenomena and understand why they are what they are. And I think in the biology of the future, we'll have to describe our systems in terms of mathematical equations. So we need a completely new breed of biologists that will be able to finally fully exploit the huge technological advancements that have taken place over the last 15 years. We have a luxury, we have more data that we can use, but we also have a responsibility, which is to develop the theories, the models, and the people that will make full use of that data.
Thank you, Michele. Morgante? Yeah, you can. You should. A lot of questions that I found some, I just write some words that we could discuss in the second part of this session. But I would like to ask to Mauro Giacca, that has a different point of view, because you have the point of view of the application and the implication of genetics and genomics in medical science and in public health. And so can you just help us, as I asked to Michele Morgante, to understand the new landscape of genetics applied to human health?
Thank you very much, Beatrice. Let me start off by really thanking the organizer for having me here and congratulating for this fantastic event that still remains fantastic despite COVID. And it is my first live event since February. And so I wish also to thank additionally for that. It's a tremendous opportunity for all of us. I like the way Professor Morgante put the problem of genetic research. I moved to King's College last year, and there is a building in central London, the Maxwell Building, belonging to King's College. And on the walls of this building, on the street, there are these pictures that show some of the moments that remain in history of science. So this is the discovery of the crystal structure of DNA, from which Watson and Crick then developed the model for DNA. But I agree with Professor Morgante saying that from the 1950s to the end of the century, basically, there's not been much progress except outside the laboratories. So basically, laboratories developed a lot of knowledge in cell biology and also in manipulation of genetic information. But the real revolution dates 20 years ago, with the completion of the Human Genome Project, which basically was a sequencing effort. A sequencing effort with, again, and this is the first problem that we encounter here, with a competition between a private institution and Celera Genomics and a consortium from government of several countries. And eventually this competition ended up with a famous conference, which was a teleconference. I'm saying this for those who are at home. Teleconference is not something that occurs only now. There's been 20 years ago, in June 2000, a teleconference between the Prime Minister of the United Kingdom, Blair, and President Clinton in the United States, who jointly stated that the property of the information on the human genome in terms of sequence should be publicly accessible to everybody. This is one of the first important questions. So who is the owner of genetic information? And the discovery of the sequence of the human genome by itself didn't cure cancer, didn't cure dementia, didn't give any clue on how to treat diseases, contrary to what several journals reported at the time. But there's been a tremendous accelerator of research. However, there were some discoveries intrinsic to sequencing the genome. The first one was that we are only made by 20,000 genes. So basically all what we do, what we think, how we are, including our physical characteristics, is made up of 20,000 genes. The number was thought to be enormously higher before, because, for example, at that time it was known that a bacterium, a very simple bacterium, has 6,000 genes. The fruit fly has 12,000 genes. And so basically discovering that you are only 20,000 was a surprise to everybody. And the second surprise was that we are speaking about the human genome, but indeed we are all different, and we differ for approximately 3 million AGCTs, so bricks that form DNA out of 3 billion. So 1 out of 1 million positions are different, and this constitutes the variability in the human species. So we are tall, we are short, we are fat, we got disease, we remain living very long, and we think in a certain manner, we behave in a certain manner because of this variation. So the variability was not expected to be that before the human genome. And this is where all the technological effort in sequencing applied to humans then becomes very relevant, because we want to know how we are. We want to discover, for example, if we are aggressive, we are optimistic, we are anxious, because this is genetically determined. We want to know our future, so we want to know if a newborn baby, or even before he is born, we want to know what might be his fate. And this fate, now it's clear that it is in his or her genetic information. A journalist before this session asked me, but so it is true that we are genetically determined to be? And I said, well, what is true is that we were wrong in the 70s when we thought that everything is dictated by our society or the experiences we have. So we have this culture by which basically the society, the experience, the education, the what happened in our life was somehow forging our future and our identity. And all these events still have an importance, but they are mediated by what we have in our genes. And understanding this remains a big black box, a big mystery. So we know that genetics is there, but how genetics works is completely unknown. We know, for example, that intelligence, as we can define intelligence by the IQ, is due to more than 100 genes, each one with a tiny influence in our capacity. But we know that genetics is there, and hereditary, for example, of the IQ is more important than education. Then all the coupling between knowing the genomic information and manipulating DNA came up to the possibility, permitted the possibility of using this information to cure disease. And this is what is gene therapy. Again, a very long travel. The first gene therapy clinical trial started in 1989 close to Washington, but it took 30 years before it became successful. But now we have at least 7 gene therapy products that are commercially available, very expensive, and this is another theme. Who should pay for the genetic treatments? Consider that, for example, to cure a boy from an inherited disease costs from $400,000 or euros to up to $3 million per treatment. Would you imagine, compared to the medicines we are used to take, paying 3 million euros for a single treatment? And so this is an enormous debate in the society. So who should bear the costs of using genetic therapies? And then something even more recent, because the gene therapy, as we have been known it, is a therapy in which we can simply add something. So basically we have a child with conditions by which there is a mutation in one gene, in 2 genes, and the 2 copies of the same gene that he has. And then we add an extra copy that is normal. This is how we know gene therapy. However, since 2013, we have available a technology, it's called CRISPR-Cas9. That's a technology that permits to directly manipulate and change the actual genetic sequence. So it changes the sequence of the DNA. And this is tremendously powerful, because for the first time in the human history, humans have the possibility of manipulating their own genetic information. And they're not restricted to do so in a body of a diseased patient, but they can do that in an embryo. They can do that in a sperm cell, in an egg cells. So they can change the actual genetic information of a species. And this, since 2013, there's been a revolution in all the genetics laboratories worldwide with the introduction of this technology. We ourselves, who are working on cardiovascular disease, we use it currently in the laboratory because we can manipulate DNA. There's been really a craze in the use of this technique. And this technique, for the first time, permits to do something else. That is, it permits to change genetic information of all species living on the planet. Our planet is fantastic because everything that's living in our planet, starting from plants to go to viruses, to go to bacteria, mammals, fish, amphibians, name everything that's living, has a genetic information based on DNA. And this is a technology that permits to change the DNA sequence. And so people have started to change the genetic sequence of any living species, plants that become fluorescent, fish that become fluorescent, fish that become mutated, mosquitoes that are genetically altered not to transmit malaria anymore. To what extent can we use genetic information to change the nature around us? We are the worst predator in our planet, and now we have a tool to change the genetic information of our worst species. And obviously, this can be applied also to humans, because we have technologies to manipulate embryos, including human embryos. We are by no means different in terms of biology compared to a mouse, for example. So we can have an embryo, manipulate the genomic information in an embryo, and think of, for example, coupling this with the processes of sequencing and understanding variation. And one can think that in the future, that's these days, we can have a boy with the color of the eyes we want, the color of the hair we want, perhaps the IQ we want, if we can change multiple genes. There are experiments in which 30 genes at a time have been changed, for example, in pigs so far. We can change genes that control performance. It's not by chance that 90% of the NBA players are black. They are genetically predisposed. Why can't we use some of this genetic information to improve our athletic performance? And other big problems, can we use that? We say, well, absolutely, we cannot do that. Well, but we are allowing plastic surgery. We have been allowing plastic surgeries in 50 years. So if plastic surgery is important for psychological reasons, is it more important to have a person who is more similar to what he thinks of himself, and so we can allow that this can be achieved instead of through surgery, through genetic manipulation? And there, the importance of this is huge, because one big question that emerges from this consideration is who should decide? Is it the scientist in his own laboratory who has the right to decide what can be done or what cannot be done? Is it the legislator? Is it the ethicist? I have a fantastic cartoon sometimes I showed to my students in which there is a ranch and there is a horse running away, and the horse has the name Science. And then there is the cowboy running after the horse, and this is called ethics. And then there is the cowboy's wife running after the cowboy, and the cowboy's wife is named law. This is a reality. Science is always more far than all the other components of the society. So how can we regulate this? I know that in the next speech there will be more debate about who should decide. And then we have the biggest problem in our humanity before coronavirus came out, still remains the biggest problem, and that we age. And aging means that several parts of our body start failing. Our arteries don't work well, we have atherosclerosis, our heart starts not working well, we have heart failure, we start losing neurons in the brain, and we have dementia, we don't see well, we don't hear well, we have arthrosis, so we have all degenerative conditions. So we need spare parts, we need to replace these parts. How can we do that? Well, for example, one possibility is to do transplantation. We take parts from a young person and we replace a heart, we replace a kidney, we replace a liver, but obviously the donors are much less than those who need the replacement part. Well, so why don't we take this replacement part from animals? We can breed pigs, for example, who have hearts, who have lungs and livers similar to those of humans, and use these pigs. We can genetically engineer these pigs to be more human-like. To what extent are we allowed to do this kind of xenotransplantation, so transplant organs from one species to the others? Or we can manipulate embryos. Now it's very easy to grow a human embryo in culture. We spoke before about manipulating the genome of these human embryos. We can do mixed embryos between a primate and a human. This has been done in the UK to see to what extent a human cells could mix with a pig cells. Or monkey cells. We can use embryos to derive cells and then make these cells becoming specialized cells, including on the brain. There is a group, for example, at Stanford University, who now cultures layers of neurons derived from stem cells, and they have for months in the laboratory, and they have an electric activity that seems to resemble that of the brain cortex, so growing a part of the brain in culture. Do these cultures think? What is thinking? We can reproduce this now studying from embryos. To what extent can we do this experiment? How can we push that? What is the future that it is ahead of us? A few years ago, I was invited in Chile for a meeting organized by the government about the impact, same topic as here, the impact of technology and genetics on the society. Very, very forward-looking government that of Chile. And this is a bus stop. So at the bus stop, there was this big question. Should we live 120 years? Because this is the set of the human species. Very, very interesting that the society is exposed, the lay public are exposed to this question. And this is a crucial question because whatever we do, there is evidence that we will never live more than 120 years. All the successes in medicine, in extending life span now to more than 80 years or 85 for women here in Italy, for example, will not bypass the wall of 120 years. Why 120 years? We absolutely don't know. We don't have any idea why there is this. We don't have any idea why a mouse lives 2 years and the human 120 when they share most of the genetic information. One big mystery. And should we address this mystery? Should we aim for immortality? For example, Google has founded a research institute called Calico in California, where the main purpose of all research groups is to try to understand the mysteries, the reason why we have a fixed date for our living. And then if you want to speak about, this is my last slide basically, of the impact of science, research, and politics in the society, then we have no better example of what has happened since February in our world. It is the impact of coronavirus, a tremendous impact that has highlighted all our weakness in all these sectors. We don't know how to govern this moment. We don't know if what has to be the relationship between science, research, politics, and decisions, public health, restriction to our individual freedom versus public benefit. We have been very poor, and this country is a special case in communicating information about coronavirus. And so this really has rendered all these questions that I have diluted in 20 years and we tend to forget, become obsessive and important in this period, all compressed. And we have shown to all of us, to ourselves, that we are really not apt to solve them.
Thank you, Mauro Giacca. You can. So, we can do many things. You can do many things. We know a lot of things. We have many information, probably much information than we can process, we can understand. So, as Michele Morgante said, we probably need a new generation of biologists and biotechnology. I feel incredibly old today because when I graduated in biotechnology, there was no bioinformatic course at the time. So I'm out of the market right now. Yeah.
I didn't study DNA and I studied medicine.
he said that they started to study DNA when he was at the university. So we have a lot of, the both of you put a lot of scientific issues, but not also scientific, because you spoke about political implication and also ethical implication. What can we do and what should we do? So if Sheila Jasnoff is with us from the we have a nice music, but let me know if Sheila Jasnoff is with us. Okay, hi, welcome, Professor Jasnoff. How are you?
I'm fine, thank you.
So welcome virtually in Trieste, and we hope that you are well and safe. And I don't know if you have listened to the two speakers, to Mauro Giacca and to Michele Morgante, from the issue that they put on the table, but we would like to enlarge the point of view, considering the ethical, the political, and the social issues, and so especially regarding your work. So if you want to help us to understand which are the implication, so the political, social, and ethical implication, we would be happy to listen to your brief talk.
Thank you very much, and I hope that they will start showing the slide. Yeah, thank you. So first of all, thank you to the organizers of ESOF for inviting me. Unlike Dr. Giacca, I'm not able to say yet that my period of virtual existence has ended. I would have loved to be in Trieste, but here at Harvard, we're still under strict orders not to travel domestically or internationally, so it is just not in the cards as yet. Nevertheless, I'm very happy to be part of this panel, and it's been a pleasure listening to you and to my two colleagues who spoke already. So I am bringing a different perspective. Dr. Giacca in particular, but I think both of my colleagues spoke about the relationship between science, technology, and society. And I am speaking of that relationship, but I'm speaking from the standpoint of a social scientist and to some degree a humanist who looks at that intersection and at that set of interactions, not from the laboratory or the clinic outward, but from standpoints in society. And I think if you turn that lens or that mirror around, the world that you see looks really quite a bit different. And that is what I want to lay out in these initial several minutes of our discussion, and then hope that we can come back to some of the points of tension and possible contradiction in the more open discussion that will follow after. So if I may continue with my slides. My two colleagues have already spoken about how they've used language like who we are and who humans are. And one of the things I want to point out that has maybe been a little bit tacit so far is that we're living in a technological age where it is not only the advances in the biological sciences that are developing, but also concurrently the information sciences and technologies. We've spoken about technology, we've spoken to some degree about computer capability. So when Michele, for instance, talks about not serial but parallel processing, we're talking about the fact that biological knowledge and biological techniques of the wet lab are coming together with very powerful advances in the information sciences and technologies. And this has been called, therefore, an era of convergent technologies. And I think it's very important if we're going to understand the opportunities, the potential harms, the consequences and implications of this era, to recall that we're not just finding out things about the genes and the genomes, but that we're in an era in which it's possible to do processing. And for instance, everybody in this audience is familiar with the European GDPR on data processing and the creation of this entity called a data subject. And what the GDPR is seeking to regulate is the processing of information about life. So I think that metaphor of processing is important to put on the table. Remember that one place where in the English language we use process is with processed foods, and we've already started a little bit thinking about genes and agriculture. Processed foods, particularly in the Mediterranean diet and in Italy, are considered a very bad thing. So, processing life, the very metaphor should make us wonder a little bit about what we're up to and what we're seeking to accomplish. So are there limits? The word limit has not yet entered our discourse this morning, but how far should we go is a serious question. And how can we know what those limits are and who decides? These are classic questions of law and political theory. Whenever you have a kind of power, who does that deciding and what are the limits on that power? These are questions that we need to ask. Now, my starting point for at least this morning's observations, and I should point out that I've been working on comparative studies of genetics, genomics, biotechnology in society for something close to 40 years by now. So I've seen a lot that happens, but my starting point for the particular thoughts I want to share with you go back to this painting, which is one of the major holdings of the Museum of Fine Arts in my own fine city of Boston, Massachusetts. And the painting has a very evocative title. It's called, in French, Where Do We Come From? What Are We? Where Are We Going? And I think that these are questions that, to some degree, have already been alluded to by my two scientists and medical colleagues, but I want to put those front and center. And I want to suggest that in order to have a productive conversation across these various divisions of science and technology and society, we have to be sure that we're asking the right kinds of questions. So we've heard a lot about what life is, and we've heard about the genes and our makeup and whether we can most recently live beyond the 120-year sort of span that people have been talking about. But I think that for people coming from the humanistic side of the sciences, the Wissenschaften, the question, what is life for? What are the purposes of it? Are equally important and have to be on the table at the same time. So, for instance, Dr. Giacca very appropriately called attention to that Chilean poster and talked about are there limits to how much we should age? But isn't the answer to that question not in the domain of what is life and why do two organisms with so much similarity in their genes have such different lifespans? I mean, obviously, that is scientifically deeply interesting. And as I approach the end of my own natural lifespan, of course, it is a question I'm interested in. But would I want another 50, 60, 70 years of life? Well, that depends on the second question. What is life for? What would I be doing with it? If, for instance, my body becomes the body of a normal 95-year-old and I'm housebound and can barely go up and down stairs and need to have somebody looking after me all the time, maybe that is not a life that I would wish to have prolonged. In fact, right now, personally, I'm not sure that I would want my teen years or my 20s years to be prolonged either, because it would mean giving up a set of things as well. So who holds the answers to those sorts of questions? And I think that the what is life and what is life for questions cannot really be separated one from the other. They're completely intertwined at all levels. Science and scientists, including the eminent colleagues you've heard today, and I know many of people of that degree of stature in my own circles of friends and colleagues, and I talk to them frequently. But scientists have this idea that because they're unlocking secrets at very basic levels, They hold the answers, and there's a kind of tacit paradigm, if you will, that I have taken to calling the sovereign science paradigm. And it has within it the following principles: that science and technology introduce new things, but they also are able to govern the novelty that they introduce, so long as there are not risks. And all the scientists I've ever known are very prepared to say that if there are undue risks, this is for society to determine. For instance, you do gene therapy, if there's going to be a risk to the life or safety of the individual, scientists say that that is a societal judgment. But once the risks have been dealt with, then governing novelty is something that science is capable of doing. Moreover, there's an understanding that downstream effects, as scientists think of them, will take care of themselves. So I can't tell you the number of scientists I've heard say, well, society was always surprised and fearful when new things were introduced. You know, look at the mobilization against cars, look at the ways in which society responded to the birth of Louise Brown and IVF. But now these things have been normalized, they've become natural, and as people accept, these days we hear a lot about the new normal, as people accept what is newly normal, they will come to accept these technological developments. All we need, therefore, is time to overcome public resistance, and then the courses of progress determined by science and technology will become plain. This is kind of almost an unspoken credo among many scientists and technologists of my acquaintance. And there are moves that science as an institution makes, and some of them we've heard this morning, by which to take over this territory and speak for it and govern. The first is by talking about moments of discovery, and in a way this panel was set up to talk about the moments of discovery. Beatrice, when you started out, you mentioned that there are these milestone points, and our colleagues have referred to some of them. For instance, the moment at which we stopped thinking about mapping and sequencing the human genome in sequence, but instead made it parallel with those banks and banks and banks of computers that Craig Venter introduced into the picture. In a way, that is a eureka moment as well. It's a moment of discovery. And if you listen to the history of science, as many journalists tell it too, it's full of these eureka moments. And in those moments, what we forget is the context in which the eurekas became possible. Science continues to make constitutional claims in my way of thinking, and I'll come back to this point, because science takes unto itself a set of understandings about how life ought to be governed, and that is a function, with due respect, of law, political theory, ethics, and many other domains of thinking that lie outside the sciences. Another way in which science asserts its sovereignty is through a phenomenon that I call ontological politics, and that is the definition of what a life stands for. Now, in Italy in particular, there's been an ontological politics that is happening outside the domain of science, and that is legislation that decided what the status of a human embryo is. Many researchers may deplore that. There are many differences across countries, but to some degree, scientists wish to claim the right to define what a thing is, and without paying attention to the context in which that thing is loosed into the world. And therefore, a new kind of game has sprung up, a game of language, in which the scientific community is very eager to say that if they bring a new thing into being, the right to name that new thing also belongs to them. So there have been parliamentary debates in a lot of countries I know about what to call life in a petri dish, you know, self-organizing cells that organize themselves and reproduce themselves. Who has the right to name these things? And I've been in many committees with many scientists who deplore the use of terms like mini-brain or even organoid because they think they don't sound right. But the point is that they deeply believe that with the right to make new things comes the right to name them, a right that in the biblical tradition was given to Adam, and it's a kind of claiming of that primary right to say what a thing is. As a result, we have a new kind of reappearance of biopower in which the biology seeks to govern who we are instead of ceding control to any other authorities. Now, all of this is happening in a market. And when you say you can scientize something, that is, you can render it into a scientific phenomenon, then you also naturalize it. You push it down to the private realm, and eventually you make it marketable. So we take for granted now that we can send our DNA off to a private company, and they then end up owning the data, processing, recombining, and so on and so forth. But this is a kind of phenomenon that is going on in society. It's not just about making discoveries, it's making discoveries in a context and with a purpose, and that purpose leads eventually to commodification and marketization. And we already remember that the first person who was put in charge of the Human Genome Project, whose name has since become somewhat more shaded than it was James Watson, no less a person than the co-discoverer of the structure of the double helix. And he was in part removed from his position through an internal politics in the National Institutes of Health because of his holdings in private companies. But he said at the time, find me any person who is not equally entangled, in this domain. So I think we have to keep that corporate sector involvement also in our minds. Is there a different way of thinking about these issues? Well, I was pleased to note that Dr. Giacca already mentioned the virtual conference in which it wasn't just me being beamed into Trieste, but Tony Blair being beamed into being in the same room as Bill Clinton. And this was the cover of Science magazine announcing that first sequencing of the human genome. But what was this betoken? I mean, why were two world leaders announcing these things and applauding the achievement? And if you listen to what Bill Clinton was saying at the time, we won't actually listen, but I'll flash the words up. You see the kind of rhetoric that was going with it. So Bill Clinton was a Southern politician in the United States and very used to Bible Belt kind of language. But this is what he said: today we're learning the language in which God created life. Now, I mean, I talked about science's arrogating moves, but of course politicians play along with that as well. I mean, why is this the language in which God created life and not, Hebrew or Bengali, my mother tongue, or the language of law, the language of constitutions? Anyway, but the point is that a divine inspiration was being claimed by this extremely talented politician to gain support for a kind of public investment in creating markets. I mean, that was understood to be what the public role was. To somebody who knows a lot about American science policy and politics, that last sentence in these paragraphs I've quoted from Clinton is very resonant. In fact, it is now conceivable that our children's children will know the term cancer only as a constellation of stars. Well, the conquest of cancer has been promised since at least 1972, so maybe it's not our children's children, but somebody else. But this is exactly the language in which the nuclear industry promised electricity too cheap to meter. And that was also in the mid-1950s, and we do not have electricity too cheap to meter, certainly not electricity that's not contributing to global warming. So it's interesting to be reminded that political leaders at the topmost constitutional levels were being implicated in this same set of developments and had a role in framing the discourse around them. This is why I say that the kinds of developments we're seeing are, in effect, matters for constitutional thinking, not only scientific thinking. And they're matters of constitutional thinking, at least at these levels. What are the definitions and boundaries of the human? Where does life begin and end? Where does non-life begin and end? What is the difference between humans and non-humans? And with that, we make classifications, and we need to reflect on those classifications. So what is an embryo made through in vitro fertilization? The answers differ, and we need to keep reflecting on these. And it has implications for the kind of legal and constitutional order we set up, our ideas of rights, our ideas of dignity, our ideas of property, to take some of the most fundamental. Now, this is something I've written about in huge detail. We don't have time to go into it in detail, but it is clear if one looks at the science policies of even leading scientific and technological countries, one finds, in effect, unwritten constitutions that give the answers to those questions. So in the US, you tend to see science and politics extremely strictly demarcated in rhetoric, in framing, in other ways, and our policies tend to be individually centered. But when you decide what a genetic's going to do for us, the answers tend to be centered on the individual. In the UK, there is a somewhat different understanding, and the networked character of family and society are bigger concerns, partly because there's a national health service, which is responsible for the nation as a whole. And in Germany, law plays a much more stringent role. This has partly to do with German history in the second half of the 20th century as well, but science is seen as serving law, not law as serving science. And in the US, it's almost reversed. So with that in mind, I will just put on the table that some of my colleagues and I, and in particular Professor Benjamin Hurlbut, who is a close colleague of mine, we have been talking for some years. This is from a commentary in Nature, for a global observatory for gene editing, because we think a different set of conversations is needed, not just a conversation among high-level scientists, not even a conversation between high-level scientists and high-level professional ethicists, but a more democratized one. And what should this global observatory be doing? It should be militating against urgent. The language I like here is the language that in our famous desegregation lawsuit, Brown versus Board of Education, the Supreme Court of the United States used. The term was deliberate speed, not just introduce everything as quickly as possible, partly to make money, but with deliberation. And deliberation, why? Because we're not in a race to solve this or solve that. With the coronavirus, maybe we are in a race, but even then, there's a question whether the richest countries in the world should buy up monopoly rights in the vaccines that are emerging. So we need a kind of slowness, a slowing down of the scientific enterprise taken in the round, with its commercial dimensions as well as its intellectual dimensions. We need a much more inclusive form of deliberation that includes excluded voices, voices from the margins, and these exclusions can be of disciplines, of geographical regions, and of social groups. And the idea of the observatory is to bring these voices in order to reframe the questions. And our idea is that through these conversations, we are likely to come to a more cosmopolitan version of ethics, one that displays what we call radical hospitality to other points of view, and understanding that there's more than one valid way to understand the topic of this meeting. How do genes and genomics fit into the remainder of the social world without which none of us will be even talking to one another? So with that, let me close off this presentation and thank you.
Thank you, Professor Jasnoff. A lot of issues. How do you feel, the two scientists here? Because Professor Jasnoff put on the table many different ethical and political also and social issues. We could start from the last one, from the observatory, because in this idea of one observatory on gene editing, and gene editing is the frontier of genetics. We have many different aspects. We have the inclusion, we have the engagement of the public opinion of citizens, but we also have a sort of regulation of gene editing, and so who can decide what you in your labs can do. So can we start from here? We have 25 minutes for a second round, and I would also like to take some of the suggestions that Mauro Giacca put on the table in his talk, and then so we try to stay in the time, and we also have 3 comments, three questions from the audience. And so,
yeah, I think one aspect that Professor Jasnoff did not touch is whether the advances that we are seeing in genetics and genomics will actually drive even more our society towards greater inequalities. Inequalities are one of the main problems we're facing today, with portions of the society that cannot get access to things that others can much more easily access. And there is definitely a danger that, especially in health systems such as the American one, which is largely private, that advances in medical science that are driven by genetics will be accessible to some, but not to all. And I think that's a real problem that we should discuss. I mean, from the point of view of what we can and what we cannot do, I, working on plants, feel I have perhaps less ethical concerns than someone that works on humans. But on the other hand, I live a different paradox. I mean, in plants and animals that are used for feeding the world, we could theoretically do with these scientific advances in genomics and with these technologies such as gene editing, many more things that we are ethically not allowed to do in humans. I mean, in humans, we can treat diseases, we can perform negative selection whenever we have a severe genetic disease, for example, through prenatal diagnostics of in vitro fertilized embryos. But there is not much more than we can do. We cannot do positive selection. We cannot make everybody taller or everybody more intelligent or everybody more performing physically. On the other hand, in plants and animals, we've been doing that for thousands of years. We've taken wild plants and wild animals and we have profoundly modified them genetically to make them more amenable to providing us with sufficient food. And these technologies could help us to do that even more efficiently, and especially could help us to reduce the environmental impact of the food production that is currently one of the big challenges that humanity is facing. Feeding ourselves is costly, not so much economically, but environmentally. And it's clearly one of our absolute needs to reduce the environmental footprint of food production if we want to make our economic system more sustainable. These technologies could help us a lot, but still it's much easier to get these technologies accepted when they're applied to humans than when they are applied to plants and animals. Why is that? In my view, it's because, I mean, whenever we have to take a decision, we make a risk-benefit analysis. And if the benefits are perceived to be much larger than the risks, then we decide to go forward. If the benefits are perceived to be smaller than the risks, then we do not go that way. When it comes to applying these technologies in medicine, oftentimes the perceived benefit is huge, and we don't think about the risks. We're saving a life. I was involved in a collaboration with Michele De Luca, a colleague from the University of Modena, who worked on a child that was suffering from a very rare genetic disease called epidermolysis bullosa that causes his skin to peel off from his body. And he treated that condition through gene therapy using stem cells that were taken from the very few areas of his skin that were still intact. We characterized the skin that was reimplanted onto the child, and the child is a huge transgenic organism. He's got more than 500 different insertions of a transgene in his body, and nobody worries about that. Why? Well, there are risks in doing this, but the choice is between the child dying in a year's time and the child perhaps dying in 20 years' time. And of course, it's an easy choice. I choose to take the risks in exchange for a benefit which is very perceivable. When it comes to agriculture, the risks are probably much smaller than the risk that child is running when we make a similar genetic modification. What people are not perceiving when it comes to plants and animals used in agriculture are the benefits of what we're doing. And so if there is no perception of the benefits, then the choice is clear. No, we'd rather not run any risk, even if that risk is minimal, because we don't see any benefit. And I think there is a need to make people aware of the fact that there are benefits, and even if they are harder to perceive, those benefits should become an important component of their food choices. When you eat, for example, beef, you're making a choice. You're eating something which is environmentally extremely costly, but you're not even thinking about that, and you're not worried about that. So if we made people more aware of the real risk-benefit balance when they make choices, not only when they make medical choices, also when they make choices on their table when they're eating lunch or dinner, I think things could become easier. When it comes to editing, I don't think the scientific community wants to be free to do anything they want to do. I think we are willing to follow rules as far as those rules are logical and scientifically based. So I think the observatory is welcome, but I would also encourage the scientific community to realize that fundamental research has to be open to doing new things, and then we have to discuss whether those new discoveries can be applied on a larger scale to real problems in everyday life. So let us not put a barrier to fundamental research, because we need to let fundamental research advance in order to have useful applications for society.
Thanks. One of the main themes is the relationship between public research and private research. Mauro Giacca, you mentioned about the cost of gene therapy, and it was 3 million, you said, the higher cost. Okay, so when we have these costs and when we have these investments, so we need a lot of money to make this kind of research. How can we balance the costs and benefits and also the implications? So how can we find a sort of balance between the need of new cures and of new drugs or new gene therapy for diseases, and the need to maintain the research open and public if possible?
I strongly believe that in this specific case, it's not much a problem of balancing, but it's a problem of greediness by the pharmaceutical industry. I'm not someone who is against pharmaceutical industry. I mean, I really think that the system has produce enormous progresses. But in this specific case, there is a problem because the cost for these new medicines is usually calculated on the cost that the same child, if it would not be treated, would cost to the system. So, for example, this specific 3 million were related to a new gene therapy for thalassemia, and the cost of a child with thalassemia for the public or the private, the insurance, so the total cost of medical care would exceed that money, and so the pharmaceutical industry think that this would be a reasonable price. This is a completely faulty way of thinking, because I mean the progress that makes, for example, gene therapy for thalassemia working comes from 30 years investment, most of which is public through public funding. It is completely shared around the world, and so basically the company that develops that final step has benefited from that, should make money that is proportional to its own investment, but not calculated in this manner. If I can add something on the previous topic, because I think this is important for discussion. I totally agree with Professor Jasnoff and also with Professor Morgante that really it should not be the scientist to decide what to do, what he can do. The scientist goes, I'm taking a term from Professor Jasnoff's slides, goes for eureka moments. What counts for the scientist is the eureka moment. I found something important. So he's not the one who should decide. It should be decided, obviously everybody would say, it should be decided by a mix of lay public, jurists, bioethicists, and politicians, and so on. But, sorry, how many politicians or administrators are here in the audience? Nobody.
Raise your hand.
But is there somebody here who believes that all of them, for example, the administrator of this city, this region, this country, are attending through teleconferences to this session? Let's hope so. Let's hope so. But this is a problem, that there
Probably
is a huge disconnect. To discuss about these topics, one must know of this topic, must know on what we are speaking. And especially in Italy, there is no interest, certainly no attempt, but probably no interest in investing in getting knowledgeable about these topics. This is a country that voted for the referendum against some aspect of in vitro fertilization, that blocked completely stem cell research in this country for 20 years, because being dismantled article after article by the Corte di Cassazione. And this is a country in which the parliament unanimously voted for stamina before it was blocked at another level. And so basically, if we want to enter into this discussion, the first thing we have to do is to improve education of people. And then we can discuss about that.
Okay, Professor Jasnoff, we have two questions from the audience that link to these points that came out in these comments. And the two questions, the first one asks, how can we have conversation about science, technology, policy, and ethics at a global level, considering that each country might have a different view on these issues? And the second one asks, given how broad application of genome editing are, do you think that we as a society will decide on the legal use of the technology on a case-by-case basis or as a general application? So there are two questions that are linked together and that are linked with the today's discussion. So.
So on the first, I want to say some things both about what my colleagues have said and also about these questions. On the first of these questions about how can you have conversations if countries disagree, well, I mean, that is the whole point of having conversations at all. And this is genetics and genomics and their uses come very low down on the scale of things that we discuss cross-nationally, where there are huge differences of opinion about what we should do. I mean, you know, think about international attitudes to the uses even of nuclear weapons, and everybody agrees that these are weapons of mass destruction, and yet, of course, we don't have full international agreement, and, you know, my country has been instrumental in even tearing down some agreements that have been made. So the point of disagreement is not to stop discussion, it's to highlight a greater need for discussion. And similarly about the broad applications and does it have to be case by case or not? Well, this has been a ongoing debate in the regulatory community about all kinds of advances in science and technology. What is the right scale? Is it better to go incrementally case by case, or is it better to approach with general principles that are applicable to entire classes and categories? My view is that there isn't a single sort of silver bullet answer to any of these things, and one needs much more about the specifics of what is going to be said. But that said, I want to turn back to some of the other things that have been said. So Dr. Morgante neatly sidestepped everything I had said by alluding to something that I allegedly had not said, namely the question of equity and equality. But there was an implicit assumption that questions of equity and equality only click in when we have the application already and it's unequally distributed and there's not enough access. I was making a deeper point about equity and inequality, and that is equity and inequality in the right to frame which questions should be governing as we create a governance structure for advances in science and technology. People care about different things. I live in a country where my political views are not consistent with the political views of some of the elements that are now in power. And these have profound consequences for things like fundamental research. And, you know, I suspect that there would be quite a lot of agreement within this community, within this committee, about how things ought to go forward and ought not to go forward. But I still rebel at a democracy theory that says that the Italian Parliament's decisions are to be overruled because they didn't get the science. I mean, may I point out that maybe the unanimous vote to support stamina does not have to do with ignorance, but has to do with the nature of the intersection between science and society, and that a degree of arrogance that says you have to learn the basics of cell biology and genetics and genomics before you're allowed to vote is maybe not the right way to go. And there were many, many reasons to be skeptical of stamina that did not have only to do with the nature of the science and technology. I mean, a deeper question may be, why did such a thing come into being? I mean, what was the nature of the desperation driving the parents that wanted to go for this kind of untested therapy? And do we as a society have a way of dealing with that kind of desperation? I mean, maybe it's science's overwhelming hubris that we have the solutions to everything at all times that actually drives people to seek alternative therapies, even when science is by no means ready to provide these kinds of solutions on demand. I mean, of course, science should progress, but the question is with what attitudes towards the rest of humanity, their capabilities, to the stringency and rigor of thought in fields other than science that may sometimes have a more accurate vision of what is going on inside of science as an institution than the people who are sitting at the bench with their narrowly defined, beautifully understood and framed problems that are nevertheless leaving out 99.x9% of the human condition.
Thank you. Okay, Michele Morgante.
Can I ask Professor Jasnoff a question?
Yeah, you have 5 minutes.
So, would you be in favor of a sort of direct democracy when it comes to science? In Italy, as you may know, we are currently governed by a party that is in favor of replacing representative democracy with direct democracy, giving people more power without any intermediation of politicians. And I mean, if you think about science, we could think of scientists as politicians. So we are the mediators of knowledge. Would you like to remove that layer and let people decide directly without any intermediation?
So this is what a lawyer like me tends to see as a leading question. And I will throw it back to you by saying that democracy theory, which in the West has a respectable 2,500-year tradition or something like that, has been deliberating this question of when is it appropriate to put something to a kind of referendum-style direct democracy and when not. It is widely known and well accepted that mob rule is one of the sort of slippery slope consequences of what might be called direct democracy. In any case, I personally am with some idea of deliberative democracy, except that that too is it's a thick theoretical area. What counts as good deliberation? Where do we do it? In what way? I mean, throwing something open to the public as a whole is something we have not done since the beginnings of modernity. There are many, many areas of our daily lives in which we simply accept that things are going to work as they work without having a clue as to what's going on, behind the scenes, and we accept it. Esau sends me a note saying, click on this link. It appears as a live link. I mean, I don't even know exactly what the mechanics of creating a live link on my computer are, but I understand that if I go through these steps, I will be, quote, with you, unquote, for this occasion and participating in some sense. So there are many places where one buys into these things, but why one does it still needs to be answered. And I think that it is not a choice between democratic control over scientific discovery and scientific progress and, throwing every decision that we make open to direct democracy. There is so much thinking about the nature of right democracy that we cannot get into that. The point is to put on the table that there are people who think about what appropriate democracy is. And to the extent that our observatory idea gains traction, and we're about to launch in 2021, I'm happy to say, we will be deliberating these kinds of questions with the participation and help of people like you. I mean, so our view is that science in society is not the prerogative of science, it's not the prerogative of society. Society is full of scientists, scientists belong to society. This has to be a multilateral, deep-going, ongoing discussion about the future of humanity.
As Aretha Franklin said in a song, you need me and I need you, and without each other there is nothing people can do. So we can find a sort of synthesis of this long session that has come to the end, and I think that the end is that we need to work together. You as a scientist, the social scientists, and the philosophers and ethical experts, and also us as a science journalist and science communicator. So we have to work a lot together to find a way to work and to improve science without making too much damage. Okay, so we try to do this. So thank you very much to Michele Morgante, to Mauro Giacca, and to Sheila Jasnoff from Boston, and thank you to our audience here in Trieste and to the online audience. Ciao a tutti, and thanks to ESOF.